early drug discovery, impurities are everywhere. You've got synthetic byproducts, degradation products, residual reagents. The list goes on. But here is the million-dollar question. How much should we really care about them at this super early stage? Getting bogged down in every tiny peak can seriously slow you down, but ignoring them completely can lead you down the wrong path. So, what's the right balance? A common trap is thinking that every single impurity you see on your chromatogram needs to be fully identified and characterized, right from the get. But in the discovery phase, that kind of thinking can be a huge roadblock. It's like trying to polish every pebble on a beach before you've even decided which part of the beach you want to build your sandcastle on. This approach can burn through time and resources for no real gain. The key is to understand the different mindsets for discovery versus development. When you get to the development stage, closer to clinical trials, impurity profiling becomes all about regulations. You have to meet strict guidelines set by agencies like the FDA. But in discovery, it's not about regulatory compliance. It's about making good decisions. The only question you need to ask is this: is this impurity messing with my scientific interpretation? Is it affecting my data in a way that could lead me to make a bad call on which compound to advance? Let's break down what we're usually dealing with. In discovery programs, impurities typically fall into three main buckets. First, you have process-related byproducts from your synthesis. Second, you get degradation products, which tell you about the compound's stability. And third, there are residual reagents, or leftover starting materials. Each of these tells a different story and requires a different level of attention. Process-related impurities, those little extras that pop up from incomplete reactions or unexpected side reactions, are the most common. In most cases, if they're just minor blips on the chromatogram, you don't need to drop everything to figure out their exact structure. You just note them and move on. They only become a problem if they start showing up at significant levels, say, over 5 or 10%, because that might signal a problem with your synthetic route. Now, degradation products are a different story. These are often much more important to pay attention to, even early on. Why? Because they're a direct signal about your compound's chemical stability. If your molecule is falling apart on the benchtop or in solution, that's a major red flag. It could kill the compound's potency, make it impossible to formulate, or signal a huge headache for future development. These are the chemical liabilities you want to find and fix early. So how do we measure these impurities? In the fast-paced world of discovery, we often use a simple method called relative area percent from a UV chromatograph. It has a quick and dirty estimate of how much of an impurity is there compared to your main compound. Is it perfectly accurate? No. Different molecules absorb UV light differently, but for making go or no-go decisions, it's usually good enough. We don't typically need to do complex response factor corrections until quantitative accuracy becomes absolutely critical for a specific decision. So, when do you need to roll up your sleeves and actually figure out the structure of an impurity? Structural elucidation becomes necessary in a few key situations. If an impurity is present at a really significant level, you need to know what it is. Or, if it's hiding under your main peak, what we call co-elution, that can artificially inflate your purity measurement. And most critically, you need to investigate if there is any chance the impurity itself is biologically active and skewing your SAA results. This brings us to a crucial point: avoiding unnecessary work. Isolating and doing full structural characterization on a minor, inactive impurity can take weeks. In discovery, that's an eternity. You should only commit to that level of effort if you have a very clear reason. Ask yourself, will knowing the structure of this 1% impurity change the decision I'm about to make? If the answer is no, it's often best to put that task on the back burner. Why is this so important? Because impurities can seriously mislead your structure-activity relationship, or SAR. Imagine you're testing a series of compounds and you see a sudden, exciting jump in potency. You think you've made a brilliant design choice? But what if that high activity isn't from your target molecule at all? What if it's from a highly potent impurity that just happened to be in that one batch? Without careful analysis, you could end up chasing a ghost, optimizing your chemistry around a false positive. There are a few analytical red flags that should always get your attention. If you see wild, unexplainable shifts in potency from one batch to another, that's a big one. Another is seeing unexpected degradation products pop up, suggesting your molecule isn't as stable as you thought. And finally, poor chromatography, where peaks are broad or overlapping, can hide a multitude of sins. When you see these signs, it's time to pause and investigate more deeply. The smart approach is to use a fit-for-purpose impurity strategy. This means you don't have a one-size-fits-all rule. Instead, you strategically prioritize. You focus your energy only on identifying and understanding those impurities that could genuinely affect your chemistry decisions, your biological interpretations, or your early read on stability. Everything else is just noise at this stage. Of course, this strategy evolves. As your program gets closer to selecting a final drug candidate and preparing for those enabling studies, the level of rigor has to increase. You'll start to shift your mindset from decision-driven discovery to regulatory-driven development. The bar for purity gets higher, and the need for full characterization becomes standard practice, not the exception. This is when you start preparing your data package for regulatory review. But let's bring it back to the beginning. In early discovery, the guiding principle should always be decision impact. Don't get caught up applying late-stage regulatory standards to an early-stage research project. Ask what you need to know right now to take the next logical step. By focusing on what truly matters, you can maintain scientific rigor without sacrificing the speed and momentum that are so critical to success in discovery. Ultimately, effective discovery is about smart prioritization. Knowing which impurities to investigate and which ones to ignore is a key skill that separates fast-moving, successful programs from those that get stuck in the weeds. Thanks for watching. In our next episode, we'll dive deeper into the practical workflows for structural elucidation and explore when it is time to bring out the heavy-duty analytical tools like mass spec and NMR. Make sure you subscribe so you don't miss it.